So, as mentioned, tonight's lecture is not going to be the one that we had anticipated. And as I said, instead we have a plan B. That's me. I actually have a hat. This was on a cruise in 2010, and we changed plans so much that I have a hat that says, the hard hat says Plan B, because that's the way it is at sea. So, for most of the work that we do, we always try to have a backup plan. So what I'm really going to talk about in what you see on your program is exploring Pulley Ridge, the deepest mesophotic coral reef on the U.S. continental shelf. And I'm going to talk about this one particular project, but I'm also going to use it as an opportunity to weave in some other things that tell you a little bit about my past in terms of some of the ocean exploration work that I've been involved with since I've been at Harbor Branch. But first, we should talk about coral reefs. And one of the reasons I was excited to give this talk in this year's series is, I talk a lot. Usually I talk a little bit about Indian River Lagoon, and then some years I talk a little bit about aquaculture, and I'd like to mix it up a little bit. And I realize I really haven't, I don't think, really talked about coral reefs. We have a number of people here at Harbor Branch that work on coral reefs. There's a number of really good reasons why we study them. They are known to be hotspots of biodiversity, so lots of organisms, lots of fish, lots of invertebrates, more so than a tropical rainforest. You sometimes hear them referred to as the ocean's tropical rainforest, but they're actually much more diverse than even tropical rainforests. They take up just a tiny fraction of the bottom of the ocean floor, but are responsible for a very high percentage of all of the biodiversity, all of the marine life that's in the ocean. So again, really interesting hotspots. And on the economic side, they're incredibly important. And we think about them primarily, in the United States that, well, we like to go to the Keys, we like to go diving, and so obviously there's some tourism monies there, and Florida is really dependent upon that. But in a lot of parts of the world, people are heavily dependent on these reefs for food. So a lot of them are fishing, but also there's some medicinal uses, and a big value of coral reefs is protection from storms. When we have large storms, it's the areas behind coral reefs that actually do better than those that are not protected by coral reefs. And you also probably have heard over the years a number of speakers in the series talk about how coral reefs are in decline throughout the world for a number of reasons. We used to spend a lot of time arguing about those, whether it was water pollution or overfishing or diseases. And the big one really turned out that it was climate change, and it was occurring throughout the world, places that didn't have all these other problems. So it's an incredibly interesting challenge for those of us who are interested in coral reefs. And as I said, we have a number of people here who study them. One particular type of coral reef is mesophotic coral reef ecosystems. And what does mesophotic mean? It means middle, light, so not too much light, but more than darkness. So enough light that we can have photosynthetic organisms like corals that have these symbiotic algae called zooxanthellae and macroalgae, things that I study. And if we look throughout the world, they're concentrated more around the tropics and subtropics. And typically they're at depths, we say the mesophotic starts about 30 meters. It's kind of arbitrary. That's about as far down as we can do with traditional scuba diving. And they can go to as much as 150 meters or more in some of these areas. And you can see, don't know whether I should use the mouse or, yeah, here's the mouse. You can see that a lot of the areas where we are right here is really a large concentration of mesophotic reefs. We've occasionally had people talk about mesophotic systems, and they talk about something called the deep reef refugia hypothesis. And all this means is that as we are worried about the loss of our shallow reefs, like the Florida Keys, it's possible that we might get some help, if you will, some relief from recolonization from these deeper reefs that are less impacted by stresses from land. They're usually further offshore. They're usually deeper. They're not as stressed. So we have some folks here that have been studying that, not much more than I have. And this has stimulated a big increase in this kind of research and also increased costs for their management and their protection. So when I first started studying mesophotic reefs, or mesophotic algae in my case, because I'm a plant person. So for those of you who don't remember, I'm a seagrass person, a plant person, work with algae. I didn't call them that. I call them deep water algae. We didn't have that word mesophotic. It wasn't quite invented yet. And when I was first here at Harbor Branch, there had been a considerable amount of work in our nearshore areas here in Florida, right off the coast here, especially down near Lake Worth Inlet up through St. Lucie Inlet. We're up here at Harbor Branch. And there had been a large number of dives done by the Johnson Sea Link submersibles. This is a very early photo of that, the way it looked in the early years. And back then, we actually had people like John Reed, who's still here, some of you heard him talk, who would lock out from the back of the submersible, and we had to do all the collections by hand. A lot of the photography was all done by hand because we didn't have good capability to collect or to photograph yet, and the submersible still worked relatively shallow. So one of the first things I did was I looked at all the data that had been assembled and went through everything and updated the taxonomy and found that in this one area alone, offshore here, there was about 208 species of algae, and 42 of them, or 20%, had never been reported before from Florida. So this gave me an idea that, yeah, there's a lot of unknown biodiversity, right here on our doorsteps. And so a lot of us over the years were fortunate to be able to use that submersible. And from that same paper, I plotted the depth distribution. So this is meters over here. So I said 30 meters is where mesophotic starts. And as we go down here, off the coast of Florida, they go down to about 100 meters, so about 330 feet. And you can see that as you go deeper, we actually get more types of diversity. And then it starts to decrease and then decreases quite a lot. And most of them, this main bar here, is actually red algae. So there's three main types of algae. The most widespread one, the most diverse, and the one that goes deepest is always red algae. And I also looked, because there have been so much collections, it's hard to imagine how many Johnson Sea Link dives went into this. It can never be repeated again because it was in the early years, and it would be unbelievably expensive now. So I think this is the only data set in the world that really shows seasonality of deep water algae. But even in Florida, we have seasonality. So a lot of us are from other places up north. We think in Florida there's no seasonality. But even in the deeper communities, we have much more diversity in the summertime than we do, say, like right now in the wintertime. So that was kind of interesting. We spent an awful lot of time in the Bahamas. So most of my work back then was done in the Bahamas. So this is my first Bahama dive a long time ago. This is a picture back then with the camera technology that we had on the wall. So the wall is when it's very vertical. They say it's better in the Bahamas for deep water algae. It truly is. It's a wonderful place to work. And my favorite spot of all was San Salvador Island. And San Salvador Island is way over here, out as far as you can go in the Bahamas, so farthest out into the ocean. And why that's important is because that's where the water is clearest anywhere in the world that we know of. And that's where algae grow deeper in the ocean than anywhere we know, 268 meters, which is incredible. So there's enough light down there at 268 meters to support growth of algae. And the coolest spot is, this is a depth profile. This is the main island of San Salvador. And off the northern coast, there's this thing called the seamount. And there's this deep area before you get to the seamount. Now, it's not really a seamount. That's why I put it in quotes. A seamount is really supposed to be volcanic. It's not really volcanic. But that's what we called it back then. We called it a seamount. And it's just this submerged platform that's in about 60 or 70 meters at the top. And when we went there, and I did this with a lot of colleagues, so that's just a close-up of where we are. This is an artistic drawing of it. What we found was an incredible, like 3D of algae. And down at the base of this is these round rock-like things that are actually crustose coralline algae. that are little rocks, little boulders, you know, about yay big. And then above them, there's layers of different types of algae that grow. And so for somebody like me, it was like this beautiful underwater garden. And this is when I got to go to 3,000 feet. Why did I need to go 3,000 feet? Because that's how deep that seamount was. And we tracked the distribution of rhodoliths on all sides, and we mapped the rhodoliths on all sides. And we did things like section them, and then did some aging studies. And we found that these boulders that on average are about 4 inches wide, that can be about 3 times that, are about 1,000 years old. So it takes 1,000 years for that much growth. It's kind of amazing. And so that was a really interesting study. And in both Florida and the Bahamas, we would see cool things. This was one that I thought I would be the one to describe it as a new organism, because I knew it was totally different than what everybody said it was. But somebody in Puerto Rico scooped me. And vertigellus literally means gooey green. So you'll still hear me at sea. I'll say, where's the gooey green? So it's vertigellus peltata. And it took 25 years from when we first got interested in this. And I had a technician here who decided to go back to graduate school, and this was in the 80s, and he said, there's this new molecular stuff that's happening with algae, and I want to understand the evolution of green algae. And so I invited him along on several cruises, and we took samples of this in the 80s and 90s. We didn't really have the right technology until much later. But when we did, let me just show you what it looks like under the microscope. All it is these little green cells, real small, tiny cells, and it's surrounded by this mucilaginous matrix, which is a fancy way of saying goo. So it's the gooey green, and very unusual species. So using molecular techniques, and I'm not going to go into great details here, other than to say you've probably seen a few of these in some of the other lectures, and basically this is just showing the relationship of all the green algae. And this particular organism is right here. And it turns out it's at the base of all green algal evolution. Who would have thought? Who would have thought? And so I never did describe it as a new species, but I did get to describe it as a new family and a new order, which is kind of a big deal. And this is my colleague, Rick Zeckman, who was that student that I mentioned who, 25 years later, this is what we did. And we actually got a really nice award on this as well. So that was kind of fun. So this is just background to say, yeah, I'm interested in this, and to put Pulley Ridge in the context for me personally. So I said it's the deepest mesophotic coral reef on the continental United States. That's kind of interesting. Where it is about 250 kilometers west of the coast, northwest of the Dry Tortugas. And basically what it is, it's a series of drowned barrier islands. So this was basically formed as a barrier island, just like the one we had where some of us may live, some of you may live, right here off our coast here now. Many, 100,000 years ago, this was where the coast was. And as time went on, the ocean rose, and successively we got some coral reef development out there. And this is what a map of it looks like. And most of it, as I said, is 60 to 70 meters deep. And it's a ridge, but it's got like 30 feet, 10 meters of relief total. So it's a very flat feature. And if you see this thing, it looks a little bit like a tie, a man's tie. This is actually the part that has been protected since 2005. It's been protected because this is a rich fish habitat, and it's called the Pulley Ridge Habitat Area of Particular Concern. And basically, it's to preserve or conserve fish and coral habitat. You can still go out there and fish traditionally, but you can't do things like trawl or use long lines, and you can't anchor, things like that. So there are some restrictions on it. So this is one of the reasons why NOAA was very interested in more study out there. And so why did I care about Pulley Ridge, though? Why was I so excited to want to get involved in Pulley Ridge? Well, it turned out that when I first came to Harbor Branch, I worked with a guy named John Ryther here. And we were working in aquaculture. And he called me in one day. He said, I got some fishermen that are saying they're finding these large pieces of green seaweed coming up in their traps. This is before the protection. And we had no idea what it is. What do you think it is? I said, well, I don't know. It sounds like, must be like sea lettuce or over or something. But it interested me. And I started thinking about it. And I looked around in the literature. And the only thing I could find was this one little paper that was written by somebody and talked about something called Anadromity menziesii. So it turns out this was first collected in 1802 and was never, wasn't even identified by a scientist until 50 years later. And it was named for Archibald Menzies, who was a very famous zoologist. I think he worked at Harvard. And it was never seen again for another 100 years after that, when some trawlers working for the U.S. Fish and Wildlife Society started to find samples. And that's when Harold Humm wrote this little paper. And that's the only thing I could think it was. And so I heard that our ship was in Texas, in Galveston, Texas, and that they had to come back to Harbor Branch. And I looked in the map and I decided that they were going to go directly over this site where I thought this plant might be. And I said, I've never done this before, but could I have like 2 days maybe on the way home? And so when I went to Galveston, sat there for a week because the seas were really rough and the previous leg had been rough. They canceled that. And after time was up, they said, too bad, we're going to go home. You're not going to get to dive. There's no more time. And it's very rough. You won't be able to do it. So as we were going across the Gulf of Mexico, we approached the site, and the person who was the operations director came to me about 8 o'clock at night. He said, you know what, we're right over your spot. It's bad, but it's not bad enough. We can launch you, but if it gets slightly worse, we're going to pull you up, and the most you can have is 3 hours, and that's it, and then we're going home. And I said, deal. So it's the first time I ever did a submersible dive, and that was in 1985. And we jumped in the water, and everyone thought I was crazy because how could I pick a spot? Back then we had no information, and I had thrown a dart. Went down, and there it was, Anadromity menziesii. That's what it looks like in the flesh. So I was the first person that ever saw it, you know, in situ, and saw a lot of other things. Then periodically I would go back. So I got some other funding to go back in the 90s, and then in 2010, some of us, I see Shirley in the room, and I think Amy, I saw some of us and a few others. We did the very last JSL cruise of our submersible following the oil spill, and we did 32 days. Brian Cousin, too, he was out with me. He and I were the only ones who did the whole 32 days. And we did some time there again. That was the last cruise of the Seward Johnson by Harbor Branch, by the way. And then the following year, we started to switch our approach, and we did an ROV cruise. And then there had also been, in the interim, some USGS cruises through St. Petersburg with submersibles and ROVs, including Sylvia Earle. Many of you know that name. And so there had been some history there at Pulley Ridge. There was an opportunity to work with the University of Miami on a giant proposal. And most of our folks here said, oh, there's no way we're going to get it. And I said, yeah, but it's Pulley Ridge. We've got to try. So that's how I got to be the lead here in what was called the community structure team. So it was one of about 6 or 7 teams organized by the University of Miami. And to my great surprise, we were supposed to have 2 cruises. We ended up with 4 cruises. To my great surprise, I thought maybe I'd go out on 1. I went out on 4, and to even my bigger surprise, I was the chief scientist for all 4. So they liked what we did the first year, and they said, keep doing it. So there was more than 30 of us, PIs. That's quite a management challenge, but that was the University of Miami. You can see Shirley right up there in the front, back here somewhere, hidden, probably back there. And we used the Walton Smith, which is a 96-foot catamaran. It's a really nice, stable platform that can work in shallow water, and it's really good for ROV or remotely operated vehicle work, which is what we primarily did. So we used the ROV to basically quantify what was on the bottom and identify sample sites for collections. There was also a technical dive part of this. The first year they came out with us and we shared the ship. After that we said, you know, it's better to have 2 vessels because it gets really complicated and we can get a lot more work done if we had 2 vessels going at the same time. So that's what we did after the first year. This is, we had 2 different ROVs. This was the one we had the last 2 years. It's the University of North Carolina, Wilmington Mohawk. I think you've seen Joshua Voss and John Reed probably talked about this in the past. And there we are working inside the ship. So basically, you know, you're inside the ship, you got a bunch of computer monitors. We have a highly skilled pilot and a bunch of us scientists trying to get as much out of the time as we can. So I showed you this earlier, this diagram. This was the focus of our first year was to work on Pulley Ridge proper. And what we did is, and we did a little bit over here on what we ended up calling the Rest Ridge, which had never really been worked very much at all. So we just did that kind of as a total exploration. And basically we divided this whole area up into little boxes, so 1 kilometer by 1 kilometer. And in each one of these little boxes, when we went to it, we did 500-meter ROV transects, and we took photographs looking down with a still camera every 30 seconds so we could quantify the bottom. And basically, we did 4 cruises, and we kind of went all over the place. So after we did our primary mission, we then worked on either side of this. Once we saw that the West Ridge was pretty interesting, we did more work on the west side, and then we ventured out into an area that initially we had called, kind of tongue-in-cheek, I never do that, but kind of tongue-in-cheek, Death Valley, because, well, these are the ridges. There's a valley in between, must not be much there. That's what everybody thought. So we ended up doing 68 of these blocks, which is quite a lot of work. And we had a total of 8 weeks at sea, 68 ROV dives, 164 hours of bottom time. We traveled 118 kilometers on the bottom. Took about 14,000 pictures, of which we used about 8,300 to quantify what we saw in terms of coverage. And it's an incredibly diverse community, is what we discovered. Our goal was to, get a certain number of images that we could quantify, for each one of these transects, and it's about 150 images per block. And we got rid of anything that really wasn't a very good photo, and we have this program called CPCE, which is a way that we can use to quantify what's there. And we have used this standardly. Again, Joshua Voss and John Reed have given you other talks on using this. And then we just tried to identify as best we could what we could see in the images. Bottom line is dominated by macroalgae. So why the emphasis is always on corals and fish, what most people tend to forget is that these mesophotic systems really are macroalgae systems, and that's why I'm so interested in it, and that's why I did what I did. So here's that Anadonium zizii again. There's also a lot of crustose species. Crustose just means very, very close to the bottom. So like lichens, if you think of lichens are crust that grow on rocks. And sometimes these algal growths really almost look like corals because they're so extensive. But they're different types of red algal crust. And I'm going to talk about the algae first because I'm an algae guy. So if you want the numbers, it's about half of the bottom, or 47%, is macroalgae, and about a third of the bottom, or most of the macroalgal cover, is in fact red algae. And this is just a little bar graph that shows you it varies overall depending on where you are. This is the main ridge, the Rest Ridge in between, off reef, and so on. But mostly wherever you are, it's red algae is what dominates. And we have green algae primarily dominating on that main ridge with that plant that I'm most interested in, Anadonium zizii, which, by the way, I didn't say this, but it's the only place in the world that it's ever been reported. So again, that tells me right there, it's a special place. There had been a previous list from some of this work I mentioned with Sylvia Earle. The person who actually did the algae is someone named Nancy Ogden, who is a phycologist who lives in St. Pete. And the list was only 27 species long, so not a very big list. From our initial, just quick and dirty, we had 29 species. With further work, we know that it's closer to 100 species, and this work is still ongoing. Red algae, so there's 3 main groups of red algae. First group is rhodophytes. Like I said, it's about a third of the bottom cover. It's about 60 species, so it's the most diverse. We have the crustose corallines that I showed you earlier, but we have these really beautiful red algae. So a lot of the algae, when you go deep, they're really beautifully, very vibrant colors. They're always very thin because when you go deep, there's not much light, and the organisms cannot invest a lot of structure into anything but capturing more light. So they tend to be very, very thin and very heavily pigmented. This is one of my favorite ones, Calamenia westii, which is easy to tell because it has all these little perforations, so it's an easy one to tell. We also have some of these crusts, and this is still ongoing, but these are called Peysonalia. And there's been a lot of work in the Gulf of Mexico that suggests there's actually a lot more species there than previously thought. On green algae, it's about 10% cover, about 25 species. So besides anadromity, some of the other notable things, I mentioned vertigellus already. It's definitely there. It's very common to find it. Codium, I'm always very interested in codium. My PhD work at University of Rhode Island was done on codium, not the species, but so you're always going to see a picture of codium, even if it's only there once. It's just my bias. And there's this little green bubble, which is also quite pretty, called Valonia. So again, a lot of interesting and unusual green species. Brown algae, or the new name nowadays is ochrophytes, only 1% cover, which is kind of small, actually kind of surprising. Only 10 species. We have things like Dictyota, which is found like in all the reefs, you know, in the Keys. It's found offshore here. It's found in the Inner Lagoon. A flat organism called Labophora. There's multiple species. We just think there's one. Again, recent work shows there's quite a lot. And a really cool one that I like to see is Spirochnus, which I first saw off the west coast here. And it's very unusual, uncommon brown alga. A lot more people are interested in the corals, and of course NOAA was. They're really funding it for the corals more so than they are for the algae, of course. And John Reed, this is really his bailiwick. We had 12 species. We had Agarecia, which is a flat plate coral. So just like the algae, when the corals go deep, they tend to be very, very flat, okay, because they're just there to capture as many photons as possible, not put anything extra into structure. Montastria, you heard Dr. Voss talk about Montastria a lot, including right here at the St. Lucie Reef. Adracus, or a finger coral. And here's some of the maximum depth distribution. So the corals there go down to, that have the zooxanthellae algae, go down to about 90 meters. So that's pretty deep. Montastria only goes to 68, so Montastria is limited to the top part of that ridge. There's a lot of other cool invertebrates, including soft corals or gorgonians. This is called Swiftia. It's very common. I see some of the sponge people in the room salivating. I'm going to show a couple sponges. Zestospongia, Utah. That's the big, large, giant barrel sponge. It's kind of to me, it's the iconic sponge in this part of the world. And if you've ever seen them underwater, you know, like I did with the submersible, you know, mass spawning of them, it's really dramatic. And here's just a few others. I'm not going to go through all of them. But Shirley's here, and Chris Diaz is up here, and that's what they do for their living. So you can ask them all the sponge questions. Fishes, we had about 86 species. Why NOAA is most interested in this is because it's such an important fishery, especially for the red grouper. And the red grouper is kind of an unbelievable thing to see in its own natural environment. We can get the estimate of sizes by using the lasers on the ROV. Those are 10 centimeters apart. So when you see lasers here, the reason we use them is to estimate size, either of the organism or how much area of the bottom we're observing in our pictures. And what's really cool about the red grouper is they're basically engineers, and they're down the bottom, and they basically make these very large pits. And they basically just go up, use their mouths, and they move pieces of the bottom around and make these very large pits, which then become gathering spots for lots of little fish. So they basically are ecological engineers. And what's happening now, though, unfortunately, and I see one of the lionfish team here, the lionfish people are in the room. When we first went in 2010, we saw a total of 6 lionfish, I think. It was the first time anybody had reported lionfish that deep in the Gulf of Mexico. They were still starting to expand rapidly throughout the Gulf of Mexico. As we did the follow-up in 2011 and did the 4 years in a row, we would go down and we would see hundreds of lionfish on a dive. Sometimes we would see 60 or 70 of these in just a single grouper pit. So where the grouper are, turns out it's a great place for lionfish habitat. And there's a very interesting story there to be told. In fact, this is a multi-beam sonar that we got from the University of South Florida. And John and Stephanie Farrington looked at this, and I don't know how well you can see it. I think you can see it. There's all these little dots here. And you can see they're kind of evenly spaced. So these ecological engineers, the red grouper, they kind of work it out among themselves. They're about every 100 meters or so. You get to see a pit. And that's where the grouper are. There's only ever one grouper per hole, if there is one. And that's where the lionfish preferentially come. So John and Stephanie made an estimate. There's 156,000 grouper burrows. That's a quick estimate how many grouper are there. And then you can also estimate lionfish from some of the work we did. Now in 2015, I don't know if Bill Baxley's here. I think he is because he called me right beforehand. And Bill was here at Harbor Branch. And, you know, people like Shirley and myself, we really can't do much if we can't collect. So until 2015, this little RV could not collect. And then with our cooperative institute that Shirley leads, Bill basically built this sampling sled that's used not just by us, but by everybody who now uses this ROV. And that allowed us to collect for the first time, 2015. So unfortunately, that was the last cruise. I wish we could have collected the whole time. Then we would have a lot more current specimens. But, you know, I could tell you about it, but really the best way is to show you a short video. And I want to acknowledge Brian Cousin, who's in the back. Brian went on 3 of the cruises with us, and we took this, he put it all together at sea, and we had a drone from the University of Miami that got the overflight of the ship while we were doing this. So that's the little ROV. It takes 3 people to deploy it. I'm usually the 3rd person in the back, which means I just hold on to the cable and they tell me to do this, and I do it, and then they tell me this, and I do that. And the other 2 guys are from the University of North Carolina. And you can see how clear the water is also. And it's not always that calm, but we always worked in August and September. And except for when we had to run away from hurricanes, which we had to do, it was a very nice place. So when you first get to the bottom, just like I saw in the submersible, this is what you see. You see a bottom dominated by this large green cabbage-looking alga, Anodontia menzii. That's a red grouper. You can see the lasers come on. You want an idea of how big that grouper is? It's a pretty big fish. You can get an idea of lots of small fish that live associated with the grouper holes. And you can see the lionfish as well. And, you know, obviously NOAA and a lot of the other agencies that we work with are really, really interested in the lionfish aspect of this. Here's some of the sponges. And more recently, Chris has been looking at these, and she can tell you more later if you ask her. But we're seeing lots of new species, lots of species that have not really been seen very often elsewhere, and, you know, new distribution records, just as we are for the algae. We had to do the fish work. We had folks from the NOAA Fisheries Lab in the Panhandle work with us each year. It was usually a different person each year. This is one of the corals, the Agaricea. This is the Madracus, the finger coral that we're zooming in on. You know, another sponge. This is the close-up of the Anodontia. I'm always interested by the little things that grow on it. Nothing seems to eat it, which is very interesting to me. There's very little herbivory down there, and I think that Anodontia is a big part of the story. Angelfish just zoomed by. More of the lionfish. And there's lots of nooks and crannies. So while it's very flat, it's actually a very complex bottom with a lot of places to live. That's that Zesto spongia again, and again, just a very rich area. One of our dives, we saw a large aggregation of urchins, and, you know, every time you go down, it's just like the inner lagoon. You never know what you're going to see. So that's my quick look with you at the video, and we have about a couple hundred hours if you want to watch those. We'll start showing that at 6. All right, can I have the next slide, Jill? Okay, so don't worry about all the numbers. What I want to tell you is just a slightly different story. This is just overall percent cover. And what I want to say is that it's dominated by algae, as I said. And that's one of the take-home messages, that it's an algal-dominated community. But in terms of species, we really have to give a shout-out to the sponges. Very high number of sponges. The numbers keep changing a little bit, but we know it's somewhere between 90 and 100 probably. We know, this is kind of a little schematic. We looked at these different areas, the main ridge to the left, the west ridge to the right. What we see is that everything is dominated by coralline crust throughout. In the main ridge, which is shallower, we have the Anodontia. In deeper water, we tend to have more of those fleshy red algae that I showed you. The hard coral is more common on the main ridge, less so as you go deeper. Montastri, which is a hard coral, is only found shallow. The agariciids are more broadly distributed, especially in the central basin. Madrocus kind of replaces them as you go deeper. The red grouper pits tend to be throughout, but especially concentrated in the central area, as are the lionfishes. And the sponges pretty much are low cover, high diversity throughout. Okay, so that's a quick summary. So we took opportunities to get some other data. Part of my story was, when I did that crazy thing in 1984, 85, I also checked what else had been done and found that Continental Shelf Associates, which is not too far to the south of us, had done some drop camera work. Back then, a lot of work was just done by putting a camera on a sled, towing it behind a ship, taking pictures. You wouldn't have any idea until you got back and developed the film what came out. And I found that they had done some sampling near some of these sites. I mentioned that to John Reed, and if you know anything about John Reed, John is tenacious. And we found, 35 years later, we found that those were still the original film rolls were in a box in a garage in storage in CSA. And John went and got them, and he came back and he clipped them all out, and we digitized them, and we looked at what it was like back in the 1980s. And then I mentioned in 2003, the USGS had gone and done some similar work with ROV. We found the exact locations where they had gone. And then in 2015, we attempted to go back to the exact locations that they had done with the drop camera in 1981 and with an ROV dive in 2003. And we were able to basically, using our technology now, we basically were able to retrace exactly where they had gone. And some of the interesting things, what about the algae? Well, the algae cover hasn't changed terribly much. It was about 55% then, and on the main ridge, it's about 58% now. And we have this further broken down, but if we just compare apples to apples, it hasn't changed terribly much. Corals peaked in 2003, about 13%. Why do I single that out? Why did we do this work? Because when we saw that number in the literature, we wanted to make sure, is that really the number? Because we don't see that now. We saw less than 1%. So what that tells us is that there's been an order of magnitude, something like 97% decline in corals at the exact same site that others had studied a dozen years before we had. And we think that's pretty interesting observation. We think that's one of the most important things we discovered. So that sounds kind of depressing, and we wondered about, what might have caused it. The answer is we really don't know. There's a number of possibilities, but none of us have the data to really know for sure. I think the simplest possibility is there could have been a disease. And again, Joshua Voss has done a lot of disease work, and you can talk to them about that. But I don't ever leave people on a downer. That's not my kind of talk. Because another amazing thing happened serendipitously. In 2014, we were supposed to be leaving Pulley Ridge and going over the Tortugas because we were also working in Tortugas. We hadn't found much, it's kind of like the same stuff. We weren't seeing anything new. And we had just enough time to maybe do one more dive. And everyone said, we should get going. I said, no, it's Pulley Ridge. Got to keep looking. So we went down, and lo and behold, we found something totally different that had never been seen before. And we were outside of where anybody had ever studied before. And it was in that area that I had been mischievously calling Death Valley, because it turns out it's Coral Valley. And it's loaded with Agaricea coral. And nobody knew it was there. And then in 2015, we went back and we really kind of documented that. And we found that it is really quite extensive. And that's really good news. And it looks like a lot of them are fairly small. And so we believe that while there may have been a die-off sometime 12 years in advance, 12 years back, sometime between then and now, we believe there may have been a significant recruitment of coral. So again, it's kind of a really interesting thing that needs to be studied a little bit more. But in a meter square, so yard by yard kind of, we found between 3, on average about 20, 17, and we found anywhere from 3 to 77 colonies. So imagine that. That's pretty amazing. And again, it was kind of just luck of the draw, if you will, serendipity, and so on. So as I said, we were one of several teams, but as chief scientist and as the scientific group that we were, our team had to do a lot of other work. So some of the stuff we did for some of the other teams was, and I had never done this before, we deployed physical oceanographic moorings. So this allowed us to keep instruments out there for, I think, 2.5 to 3 years, where we could continuously record things like temperature and salinity and currents, which is really important to understanding the circulation of the system. And so we deployed that on the very first cruise and then recovered them on our way home the last year. And so this is led by a physical oceanographer at the University of Miami. I'm not going to go into their work very much, but these are basically just showing current at different locations, at the 3 locations, and surface and near bottom, and basically their interpretation of this. The whole idea of our project was to try to relate Pulley Ridge to other coral reef systems in the Gulf of Mexico and specifically in the Florida Keys. And from a physical oceanography perspective, the conclusion of the physical oceanographers and their modeling efforts is that these systems are all physically interconnected, and it's controlled both by local and regional circulation. So a big driver, for example, is the loop current. And some of you may know about the loop current. The loop current is out in the middle of the Gulf of Mexico. The loop current normally brings water this direction. When we had the oil spill, the loop current didn't do that, fortunately for Florida. And that just goes to show you how important the currents can be in another application. So here's just some of their models that they have made. And Pulley is strongly influenced, like I said, by the loop current here. And also, local connectivity is impacted by eddies and meanders of the Florida current. And so their recommendation is that when we try to manage these reef systems, we need to take into account the neighboring and upstream reefs. So the northern Gulf of Mexico, and some of you have heard Josh talk about the flower gardens, Cuba, which we'll mention again briefly, and the Caribbean. And that we're going to need international collaboration to really fill in the data on things like the physical oceanography. There was another group called the Population Genetics Group, and I mentioned that we did collections that last year with the ROV, but most years it was done by a technical dive team. And these folks came up with a list of target species. So there was 3 species of coral, a couple of species of fish, and the Zestospongia was the sole sponge representative. And basically, they used collections of these from Pulley Ridge and from some of the other reef systems that I've mentioned, and others some that I haven't, to try to get a better understanding of the biological connectivity. So we had physical oceanographers doing physical stuff. We had the biological folks using these molecular tools. And generally, again, I'm summarizing other people's work, but generally the fishes that have a very long pelagic life duration, so their larvae stay in the water column a long time, like 3 to 7 weeks. They show really high connectivity. So for example, that red grouper, it seems to be all connected all the way up along this coast of Florida. So all throughout the Gulf of Mexico, Pulley Ridge, all around Florida, further north, which is pretty interesting. Their conclusion was that Pulley Ridge has the potential to act as a refuge for this important fishery species. And again, they say we should be managing these reefs as a part of a regional management plan. Other things like corals and sponges have shorter larval durations, so sometimes it's only a couple of days. They tend to have different population structures and lower connectivity. Josh just had a PhD student, Mike Studeman, finish up his PhD. He did a really nice project, and one of the things he looked at in terms of corals is he basically looked at Montastria and basically concluded that Pulley Ridge is not connected to any of these other reefs. So again, very interesting thing, very interesting about Pulley Ridge. And there's also some population structure based on depth. The closest thing for most of these, like the sponges and so on, seems to be a tie to the Florida Keys, not to the rest of the Gulf of Mexico so much. And again, all this data coming together saying that we need to be thinking much differently in terms of how we manage our resources. It needs to be done on a much more regional scale with a lot of collaboration, including international partnership. So briefly, what do we know now that we didn't know before at Pulley Ridge? Well, we had this great decline, more than 90% loss of coral in the past 10 years. But we also found a place that had never been seen before, the largest concentration of coral outside the protected area. So one of the things that we did is to recommend that protection. We also showed the explosion of lionfish populations on Pulley Ridge since we first were there in 2010. John Reed, many of you know that John has done an awful lot, starting with the Oculina reefs here, some of the deeper reefs offshore, the Ophelia reefs. He immediately put together, while we're at sea, the idea to expand the protected area from just this area to include this whole area here. And so that's under review now for further protection. So the science that we do on this project, we immediately try to translate into management activity. And people like Josh and John are very good in terms of working with NOAA and some of the fishery management councils. Take-home messages for the algae. Well, they're really important, but we don't know enough. They're very poorly known. Not many people study them. Their cover far surpasses anything else on the bottom, so we should probably do a little bit more work on them. We know they're important for primary producers, but we don't really know how much, the quantity of that. And there's probably a lot of other things that we need to do to better understand what their roles are at these great depths for algae, such as how productive are they, what about their habitat value, who eats them, what are their roles in nutrient cycling, what about climate change, how are they responding. So another really cool thing is we were asked by NOAA to provide them with website material. That was another one of my jobs, kind of wearing my education hat. And I said Brian Cousin was out with us, and we were able to get really good photography and some videos, including the one that we showed you. And so if you want to know more, you can just say Ocean Exploration Pulley Ridge, or you can say Pulley Ridge Connectivity, or you can just go to the Ocean Exploration website, and you can see tons of stuff, including like logs that we sent out whenever as most days we said, here's what we're doing, here's some cool things we saw, here's our new discoveries. So that's how we announced, for example, the discovery of our coral reef find. So you all know this book. So, at some point you get to go to a lot of cool places. I've tried to show some of them. It's not really a seamount there, but it kind of looks like it. And so, what I learned is that the marine biodiversity of these systems in Florida, the Bahamas, and Caribbean where I've been, it's really incredible. We're just really starting to understand it. But also, we're challenged because we're losing it before we understand its complexity. And, years ago, I didn't think too much about nature and how it was impacting things. But almost all our work on nature, we have to factor in the human element if we're really going to understand what's happening before our eyes. So I mentioned the need to go to Cuba, and I think a lot of you know we did go to Cuba. It took a lot of work, and there was a 4-week-long cruise. John Reed was the chief scientist. This was done under the CI that Shirley leads. And you can go to this website, same website, just the Cuban expedition, because we were again asked to do that, and so I did that as well. And Shirley is going to give a lecture on our Cuba work in January, so I invite you to come back and hear Shirley's most excellent talk. And we actually, some of us, including Chris, myself, and Shirley that are in the room, and Ming Sheng's in the room too, I see. So 4 of us that went are in the room, and Josh and John are traveling right now. But we were in Cuba last week with the idea of looking at the data we got from our Cuban colleagues, and we're trying to work with students that are going to be trained to work on mesophotic reefs, and we're trying to get a group of those Cuban students to come here and work at Harbor Branch for additional training and as part of their PhD work. So we're going to have quite an impact, all because of mesophotic reefs. And I'm kind of proud to say that half of those 8 students are all going to work on algae, because that's what's the coolest part, right, Chris? All right. So I got to thank an awful lot of people. Our funding, which I mentioned, a lot of this is with the CI. But the main funding for this, the Pulley Ridge, was in collaboration with the University of Miami, which also has a CI, so it was a CI kind of thing. And I mentioned the University of North Carolina and their great crew, and the Walton Smith captain and crew. It takes an awful lot for the ROV. It's not simple. The captain who's handling the ship and the ROV crew have to work very closely together along with the scientists. John and Stephanie did all of the initial photo analysis as well as doing all 4 cruises with me. And then recently, Holly and Kate have been working with me to take the algae kind of like the next step, and also 2 colleagues from the University of North Carolina. So we're basically what we're trying to do is tease out the algal side of this a little bit more. Because again, this is probably now the most extensive survey ever of a mesophotic system, especially on the, some of the things like the algae and sponges. So it was a lot of work, but it's going to have a lot of impact beyond just the immediate study. And I think with that, that's the last time I saw Pulley Ridge. So any questions?